My name is Sara Knaack, and thank you for attending this ISMB 2022 poster presentation on dynamic regulatory module networks for examining context-specific gene regulatory networks. Changes in transcriptional regulatory networks significantly alter cell function. Multiple studies have profiled transcriptomes and epigenomes during a developmental or other biological process, and systematic methods to integrate such data are lacking. To address this challenge, we present a novel approach, dynamic regulatory module networks, DRMNs, to predict regulatory networks in a cell type or time point specific manner. In this poster presentation, we highlight results from two of several data sets studied in our recent work. The first is the human H1 embryonic stem cell differentiation dataset of Xi et al. The second is a parallel RNA-seq and ATAC-seq time course dataset in Medicago roots under treatment with lipotrietoligosaccharides or LCO compounds for 24 hours. Such compounds are known to be an early biochemical signal in establishing nitrogen-fixing symbioses in plant roots, and this experiment emulates early transcriptional response in that process. DRMNs represent the regulatory network as a set of expression modules, one set for each condition, together with their upstream regulatory programs. They are inferred from input expression and epigenomic profiles using multitask learning. A gene's membership in a module is determined by the chromatin state of its associated promoter in terms of both accessibility and/or chromatin marks, and the accessibility profiles of nearby motifs in a fused lasso regression framework, which infers a regulatory model of expression for each module. This approach enables systematic integration of RNA-seq, ChIP-seq, and ATAC-seq datasets. The chromatin accessibility profiles of known motifs close to gene transcription start sites are used in the regression to represent regulator activity. From such results, we obtain per-module network predictions across a series of conditions, enabling detailed analysis of dynamic regulatory processes. Shown here are example results for the human stem cell differentiation dataset of Xi et al. At left are scatter plots showing the degree to which variation in expression is captured for each DRMN module by comparing predicted and actual expression levels. And on the right is a depiction of the inferred expression modules and differentiation dynamics of the 5 cell types studied in this dataset. Downstream analyses facilitated by the results of DRMN include benchmarking comparisons to regulator sets and networks inferred from outside sources, providing important validation for our results. Additionally, results are readily compared across conditions, shown here with the example of module gene set comparisons across the 5 human cell types studied in this dataset. Finally, the prioritization of regulatory features is of great interest. Shown here are regulatory features identified as important for genes in modules 3 and 4 of this same dataset. As mentioned, we applied DRMN to multiple datasets, including a time course dataset on Medicago roots in a study focused on nitrogen-fixing biology. Shown here are inferred modules of gene expression from that dataset, as well as the similarity of the inferred modules across time, indicating general reorganization 2 to 4 hours after LCO treatment. Noting this, we identified regulatory features presenting significant variation at this time frame. Among the implicated regulators, some have known importance to nodulation biology like Cyclops, and others are of more novel interest such as ERF1 and EIN3. We also seek to study regulatory dynamics on a per-gene level. The inferred module assignments of genes are used to cluster similarly varying genes. We applied a regularized regression model, MTG-Lasso, that accounts for grouping structure to find regulatory features that are significant on the per-gene scale of each cluster, defining a per-gene regulatory network. Four such example regulatory features are shown here for a gene set associated with gene ontology enrichment for defense response, consistent with the theme of symbiosis. From this analysis, we again identify regulators of interest, including many implicated at the module level, but some are specific to this analysis, like CAMTA1 and NIN, defining a refined picture of gene regulation in this system, also represented here with the top 1,000 predicted edges. In short, we have applied DRMN to major bulk multi-omic data sets in analyses that integrate expression and chromatin accessibility, also chromatin mark data, identifying patterns of coordinated change on both global and gene-specific scales in each of these biological systems. We also infer regulators that dominate changes in transcriptional activity and obtain target predictions for these regulators from downstream analyses of DRMN results. Future and ongoing efforts include to experimentally validate predicted regulators and targets of interest in these systems. Thank you to the ISMB 2022 organizers for this opportunity to present our work. We look forward to entertaining questions and discussions about this work, including in person at the poster session on Wednesday, the 13th of July, 2022. For those following virtually, we'd also like to particularly acknowledge the following collaborators and funding agencies for their support of this work.